The next thing that these researchers wanted to do was they wanted to check out the biases in the type of mutation that are most often seen in the biological world. Because the previous paper, the authors did not take into account the biases in the type of mutation. They figured that all mutations were equally likely. And finally, the third thing that they wanted to do in this paper is that they wanted to look at the biases in the location of the mutation. Now, note that each codon has 3 different bases, and so for an error to happen in the first base or in the third base, those are errors that are much more likely to occur than an error in the second base in the codon. So let's go ahead and unpack this paper. So the first thing they did is that they used more computing power. So in the previous work, the authors only looked at 10,000 different code variants, different random genetic codes. And in that work, they found that the natural genetic code was 2, remember 2 were better out of 10,000. Now the problem with this number is that when you only have 2 that you find that are better out of 10,000, 2 is a small number, and there could be this could be the result of a statistical fluctuation. In other words, maybe you just got lucky or unlucky, and maybe on average you should expect only 1 to be better out of 10,000. Or maybe on average you should expect 3 to be better out of 10,000. And so what you want to do is you want to have larger numbers of random genetic codes to test the natural genetic code against, to see whether if we did 100,000 instead of 10,000, whether or not there will be 20 or 19 or 21, which would basically be the same as 2 out of 10,000, or if you're going to get 10 or 30 or 5 or 50, which all would be within the statistical fluctuation range of 2 out of 10,000, since they were choosing these things randomly. And so what they found is when they looked at 1 million different genetic codes, 1 million, how many were better? How many were better out of 1 million? How many were better? And there were 114 were better out of 1 million, which means that 2 better out of 10,000 was an overestimate. They really should have gotten 1.14. Of course, you can't do that out of 10,000, which is why they needed to do a larger number of random genetic codes to get these better numbers. Okay, so this is basically the same result that they found before. 2 is close to 1.14, and so they validated their previous result while making it more precise. Okay, so the next thing that they wanted to do, lost my spot. The next thing they wanted to do was they wanted to look at a bias in the type of mutation that you can find. Oh, before I go to that, you should see this plot right here. So when they did 1 million of these, and they scored the different polarity value here, which they called the MS value in this next paper, how many different codes did they find at these different values? Well, of the 1 million codes that they found, which adopted this sort of distribution here, this 5.19, if you recall, was where the natural genetic code fell. Okay, so back to the idea of the bias in the type of mutation. So these researchers recognized that some mutations occur more frequently than others. So you have 2 different kinds of mutations, or mutations, single point mutations, are typically categorized 2 different ways. One kind of mutation you could have is a transition. And a transition mutation would be an A being mutated to a G, or G back to A. And the other kind of transition would be a C being mutated to a T, or a T to a C. So these transition mutations are by far more common than the second type of category of mutation, which is called a transversion. And a transversion would be an A or G to / from a C or T. So transitions are on these blue arrows here, and transversions would go on the diagonals or vertically. So these would be the transversions. And they're far less likely to happen than a transition. And perhaps the reason why is if you look at A, adenine, it's this double ring structure, and guanine is also a double ring structure, so it's easier to do this kind of mutation where you maintain the double ring structure. These kinds of double ring structures are called purines. And the other kind of nucleobase is a pyrimidine, and the C and the T are both single ring structures, which is why they're pyrimidines. In other words, the takeaway message here is that they needed to score their random genetic codes by these transition mutations being more likely than their transversion mutations, right? So that's how the bias in the kind of mutation would affect the results, right? So what you have is if penalties of transversions are higher than transitions, then the original calculated score is artificially inflated. So what do I mean by that? Well, I'm not going to go through all of the calculation, but if we return to the example problem that we had at the beginning, what we did is that we looked at every single one of these 9 different ways that UUU could be mutated by a single base pair mutation. Right? So a U is the RNA equivalent of a T, right? And so this U being transitioned to a C, which would be this one here, and this one here, those 3 kinds of mutations are far more likely to happen than the other ones. And so what the authors did in this second paper is that when they calculated their average change in polarity, they calculated it on the basis of having this transition mutation being weighted more heavily than these other ones. So they calculated these weighted averages, and so they didn't count these penalties being these deltas over here, as much as the ones for the 3 transition mutations. Now, as you can see, oops, in this particular case, that penalty actually is a little bit higher if you weight these 3 more, because you have the by far largest penalty occurring on a transition mutation. But that's okay. They weren't trying to bias their results. They were just trying to see what would come out if they did this kind of research. So what I meant by this sentence is, if you look at this at the table of the penalties that I just showed, those deltas, if the penalties for the transversion mutations ended up being higher than the penalties for the transitions, but the transversion mutations are less likely to occur, then the score that you calculate is higher than it should be if you weight the different kinds of mutations properly. Now, the second kind of bias that they looked at, and to jump to the punchline, yes, this ended up actually, when you took into account this bias of the kind of mutation, then it did actually improve the score of the natural genetic code, which we'll get to in a little bit. But the second kind of bias that they wanted to investigate was the bias in the location within the codon. So if you imagine a mutation happening to the DNA itself, the bias, there would be no bias in where the mutation might happen within a codon. But if you're looking at a kind of mistranslation, so if you have your RNA, as we saw before, and there was a UUU on your RNA, and that was trying to pair with an A anticodon on your tRNA. So this is your tRNA, and down here is your mRNA. Right, so this codon would code for phenylalanine. There's a possibility that instead of the AAA anticodon tRNA coming, you might get instead a CAA anticodon coming to pair with a UUU. And that would be an error in translation. Now there are the mismatching here in the mistranslation is going to occur more frequently in the first base of the anticodon, or maybe I should say in the third base of the codon, or in the first base of the codon, but very, very, very rarely would it occur in the second base of the codon. In other words, the error frequency of the three different positions within the codon is your third base is more likely to have an error than your first base, and those two are far more likely to have an error than you would in your second base in the codon. Furthermore, they wanted to look and see what kinds of biases in the kind of mismatch you would find depending on where you are in the codon. So second codon errors, in addition to the second codon error being the least frequent happening, the second codon errors are almost never transversions. In other words, in the unlikely event that you do have a second codon error, that is almost always a transition. Next, first codon errors, heavily biased to transitions. So perhaps even more biased than normal, but not quite as much as second codon errors. And third codon errors, they have little bias. So they wanted to look at both what is most likely to occur to your DNA, as well as what would most likely happen when you have a mistranslation. And so when they take into account both sets of biases, how does the natural genetic code fare? Well, it turns out that when you take into account these biases, only one code out of 1 million randomly selected codes, again with the same structure as the natural genetic code, randomly selected codes was better. So only one was better than the genetic code out of 1 million when you take into account all these different kinds of biases.